Okay, imagine this. Everything alive, like the plant on your desk, maybe even you, is subtly glowing with a light.
Yeah,
But it's so faint we can't see it. Sounds kind of like science fiction, right?
it really does. But, for decades, scientists have had this hypothesis, this idea of ultraweak photon emissions, UPE, or sometimes called biophotons, coming from living things.
No.
And now, well, there's this study that seems to provide really compelling evidence. It looks like it confirms it.
Right, and that's what we're diving into today. Our main source here is this recent paper by Solari and the team. The title is Imaging UPE from Living and Dead Mice and from Plants Under Stress.
Yeah.
So we're going to try and unpack what these emissions actually are, the sort of scientific journey to finally detect them.
Which wasn't easy.
Right. And what these experiments, on mice and plants actually showed us. And honestly, what this all might mean for how we understand life.
And look, let's get something clear right away. This is pure science we're talking about. It's absolutely not anything mystical, no auras in that spiritual sense, none of that.
Like what?
This is biochemistry, it's electromagnetic emissions, just incredibly weak. It's also not like firefly light, not visible like that, though the principle is related. And it's way, way weaker than the infrared heat your body gives off, the kind thermal cameras see.
Right. Ultra weak is definitely the key phrase here. So, okay, diving into the science then. What is this UPE? Like fundamentally, what kind of light is it?
So it's optical light, which means technically it's in the same spectrum our eyes could see.
Oh, wow.
But the intensity is it's astonishingly low. Billions of times dimmer than, say, the light in this room.
Billions, wow.
Yeah, think of it like stray photons being released. Just a few here and there. So few that you absolutely need incredibly specialized, sensitive equipment to even pick them up.
You mentioned this idea, this hypothesis has been around for a bit. Was it always accepted or was it more on the fringes?
Oh, very speculative. For a long, long time. Yeah, the whole concept of biophotons goes back decades. People tried to detect them before, but, well, it was tough. The theory was often linked to chemiluminescence.
Like chemical reactions making light.
Exactly. Like molecules get excited, chemically speaking, and then they release a photon, a particle of light, as they sort of calm down.
Okay.
Fireflies do that, but the biophoton idea was that this happens on a much, much smaller scale, probably tied to basic metabolic stuff, like involving oxygen, proteins, oxidation reactions, that sort of thing.
So the theory made sense chemically, maybe, but the proof was just hard to get because the light was just too incredibly dim.
Precisely. It was seen as pretty controversial, definitely unproven for many years, because reliably detecting something that faint was just technically super challenging, maybe impossible with older tech.
Okay, but the theory was a bit more specific than just chemical reactions make light, wasn't it? There was a proposed reason why cells might emit this light, especially when they're stressed.
Yeah.
How did that work?
Yeah, the main theory connects UPE directly to cellular stress. So when a cell is under pressure, could be UV light, chemicals, maybe lack of food, whatever, it can start producing these things called reactive oxygen species, ROS.
ROS. Okay, I've heard of those, like free radicals.
Sort of, yeah. They're highly unstable molecules. They react really quickly with other stuff in the cell, like proteins or fats, lipids.
Okay.
And when ROS reacts with these things, it causes oxidation. And in that process, electrons inside the molecules get sort of bumped up to a higher energy level. They get excited. And when those electrons eventually fall back down to their normal state, they have to release that extra energy. And how do they do it? By spitting out a photon, a particle of light.
Okay, that makes sense. So if that's the mechanism, stress leads to ROS, ROS leads to oxidation, oxidation makes light. What's the big deal? What's the potential significance if that's really happening?
Well, this is where it gets really, really interesting, potentially. If the light emission is a direct result of cellular stress, then detecting that light could let you literally see physiological stress in a living thing.
Whoa.
You could potentially see which cells or tissues or maybe even a whole organism is distressed just by seeing where this faint light is coming from.
Like a glowing map of stress or disease.
Exactly like that. I mean, think about the possibilities. In medicine, maybe detecting diseased or stressed tissues non-invasively.
Without cutting someone open.
Right. Or in agriculture, maybe spotting stressed crops way before a farmer sees the leaves wilting. Or animal health, monitoring how livestock are doing.
So this huge potential was there. The theory was kind of making sense.
Yeah.
But the breakthrough, according to this Solari study, really came down to the technology, didn't it? What kind of camera finally made this possible?
Yeah, the tech was crucial. The key thing highlighted in this study, which came from a team at the University of Calgary in Canada, was using these incredibly sensitive cameras. They're called EMCCDs, electron multiplying charge-coupled devices.
EMCCDs.
Got it. These are not your phone camera, not even close. They're designed to detect and actually amplify the signal from even a single photon hitting the sensor.
Wow, single photon.
Yeah. And that is what finally allowed them to reliably measure and actually get images of this incredibly weak light that was basically invisible before.
So they had the magic camera. How did they set up the experiments to test this on, actual living things? Let's start with the mice. What was the setup and what did they find?
Right. So they used 4 mice in this part. They were anesthetized, kept warm on a heating mat, and then, well, they were euthanized. The EMCCD camera was focused on them the whole time, capturing any optical light being emitted.
Yeah.
And the finding was pretty definitive. The amount of optical light they detected coming from the mice dropped dramatically, and quite quickly, actually, within about an hour after they had passed away.
That seems pretty conclusive. The light was there when they were alive and metabolizing, and it basically vanished when those life processes stopped.
Exactly. It strongly suggests the light was really being generated by processes happening only in living cells. But they didn't just stop with mice. They also did experiments with plants.
Okay, how did that work? With leaves?
Yeah, they took leaves from a couple of different kinds of trees, and they applied chemical agents to deliberately stress specific spots on the leaf.
Ah, okay, inducing stress.
Right. And using the same super-sensitive camera, they watched what happened. And in the regions where they induced the stress, there was a clear increase in light emission. The stress parts started glowing brighter in this UPE sense.
So the opposite of the mice stress made the light increase in the living plant tissue.
Precisely. And this brighter glow lasted for many hours. But interestingly, it gradually went back down to normal levels after about 16 hours or so.
As the leaves recovered.
Presumably, yeah.
As the leaves sort of dealt with the stress responses calmed down, ROS production likely stopped, and the light emission went back to baseline.
Wow. So a drop after death, an increase with stress, and then back to normal. That's quite a story. It is. And they also noted something interesting. When they used benzoin, which is actually a local anesthetic on the leaves, that seemed to produce some of the highest light emissions.
Huh. Why would an anesthetic cause more light?
Well, it suggests the link isn't just stress in a general sense, but it's tied to specific physiological states and how cells react. Different chemicals, different stressors, maybe different physiological changes could produce different light signatures, which again points towards that potential diagnostic use.
Okay, so pulling it all together.
Yeah.
These experiments, the mice, the plants, using this cutting-edge EMCCD camera.
Yeah.
What's the final verdict? Is the hypothesis confirmed now? Do biophotons exist?
I'd say these experiments provide very, very strong evidence, almost definitive confirmation, really.
Yeah.
Seeing the light drop after death, seeing it increase with stress, it makes a very compelling case that, yes, living things like mice and plants do emit these ultra-weak photon emissions, these biophotons.
We're talking what kind of intensity, again, just to remind ourselves how faint this is.
We're talking maybe thousands of photons per square centimeter per second, which sounds like a lot, but compare that to the billions or trillions hitting your eye from daylight, it's incredibly low. But it's definitely there and detectable with the right instruments.
So, okay, we know it's real, and we have a lead of theory about why it happens linked to cellular stress and ROS. But I'm guessing the study didn't claim we have all the answers now, right? There must be remaining questions.
Oh, absolutely. Confirming something like this just opens up a whole new batch of questions for researchers.
Like what?
Well, the study itself brought up several things. For instance, while the ROS and oxidation mechanism is the leading idea, they ask if other parts of the cell, maybe mitochondria, are directly involved.
Mitochondria, the cell's power plants. Why them?
Well, mitochondria are key to energy metabolism, and they're also known to be major sources of ROS, especially when the cell is stressed. So it seems like a really plausible connection to investigate further.
Makes sense.
They also speculated on some pretty intriguing, maybe more out there ideas, like could other maybe animals with different kinds of vision, like cats perhaps, could they potentially see this light?
Whoa, cats seeing biophotons, that's wild.
Yeah.
But just speculation at this point.
Yeah, definitely more on the speculative side mentioned in the source material, not something they tested, just what if.
Okay. And you mentioned the link to oxidation earlier. Could that tie into things like blood flow, delivering oxygen?
That was another specific question they raised, yes. Since oxygen is pretty crucial for those oxidation reactions that seem to generate the light,
Right.
they suggested experiments to see if changing blood flow, and therefore oxygen delivery, directly affects the UPE levels. It would help connect the dots between the tiny cellular mechanism and the bigger picture of the organism's physiology.
It really does highlight that, okay, we've confirmed that life glows faintly, but the exact how, all the molecular details, how it all fits together, that's still being worked out.
Precisely. The confirmation is a huge step, no doubt. But it feels like just the start of really understanding what this phenomenon is all about.
Yeah.
And as we keep saying, none of this discovery, none of this confirmation would even be possible without that leap in technology. Those incredibly sensitive cameras really were the key.
So, wrapping it up, we've moved from this old kind of controversial idea to actual scientific confirmation, largely thanks to tech letting us see something completely invisible to us.
Yeah.
Living things, from a humble leaf to a mouse, genuinely emit this incredibly faint, subtle light.
It's a great reminder, isn't it, that even in systems we think we understand pretty well, like basic biology, there can be these fundamental processes going on at levels just completely beyond our normal senses.
Wow.
Processes that only show up when we build the right tools and ask the right questions.
And the potential implications, still need tons more research, but they are genuinely exciting. Thinking about visualizing stress, maybe disease, new ways to monitor health and farming, animal care.
It definitely shifts how you might think about the subtle sort of energetic landscape of living systems around us.
So here's a final thought for you, something to maybe mull over after this deep dive. We've just seen how science confirmed that life, all around us, is subtly glowing in a way we literally couldn't see before. What other fundamental unseen signals might living things be emitting right now? Signals we haven't even thought to look for yet, let alone started to understand.